All right, folks, let's get into the biochemistry of it all, which is going to be our focus of macromolecules. So there's 4 types of macromolecules, and I'll be showing you a chart to help you kind of organize everything as well. And a similar chart like this is going to be in your chapter 2 worksheet. Okay, folks? Now, the 4 types of macromolecules are going to be our carbohydrates, our lipids, or our fats, our proteins, and our nucleic acids. And the specific thing about any of these macromolecules is that they must contain carbon and hydrogen. Okay, folks? So all of them are going to contain carbon and hydrogen. Some are going to contain oxygen. Some are going to contain nitrogen, sulfur, phosphorus, etc., etc. Now, when it comes to these organic molecules, a lot of them do have their own building blocks. So carbohydrate, the building block is going to be a small glucose sugar. For proteins, the building blocks are going to be amino acids. For nucleic acids, the building blocks are going to be nucleotides. And we've seen a nucleotide when it came to the adenosine and adenosine triphosphate, or ATP. We saw the amino acids when we talked about synthesis reactions, and we saw the glucose when we talked about mixing sugar with water, for example. Okay, folks? And these monomers are like the small parts of a carbohydrate, a small part of a protein, a small part of a nucleic acid. And when you have a bunch of these monomers coming together, you form a polymer. So monomer, mono means one, mer means part, so that's one part. Poly means many, and again, mer means part, so that means many parts. And that's the beauty of macromolecules. You can build them up, you can break them down, and here's a different reaction to help do that. You have a dehydration synthesis. Synthesis is the key word that we saw from one of our chemical reactions. That means we're going to be building something up. So you take a monomer, and you're going to be forming water. So that's where you have a little arrow to show that water is going to be released. And when that happens, now these monomers are linked, and you are now creating a synthesis. You're building things up. And this can continue to happen as you add more and more monomers to be attached. Then you have hydrolysis. So remember we were talking about how we can go with a dehydration synthesis in part A. Well, if we were to do the reverse and do a sort of a decomposition, that means we can do something called hydrolysis. So hydro means water, lysis means to break. So it means you're using water to break a bond. So for example, you have 2 monomers that are linked. You're going to be adding water, and now you have 2 separate monomers as product. So this hydrolysis is similar to a decomposition reaction that we saw before. So here's an example of dehydration synthesis and the breakdown by hydrolysis. And the beauty of this type of reaction that we see here is that we see 2 arrows. That means we have an arrow going forward and an arrow going in reverse. When you see 2 arrows, that means things are in equilibrium. So you can have a forward reaction to form these products, and then these products can have a reverse reaction to form these reactants all over again. So it's almost like you're forming products and reactants over and over until they reach an equilibrium. So let's go with the forward reaction. As you can see, we're taking 2 monomers and creating a disaccharide, or a little version of a polymer here, which is 2 sugars. And then if you want to add water to this, to go into the reverse, we can break it back down into its individual monomers. And that's the beauty of it. Now, one thing you might be saying is, where are the lipids at? Where are the lipids at? Lipids are a little bit different because they're made of different pieces, like triglycerides. You can have phospholipids and steroids. And so because of that, they don't have one little perfect monomer like you would see for carbs, proteins, and nucleic acids. So we just kind of talk about those a little bit separately. But here are macromolecules, and I want to get into carbohydrates and lipids and kind of show you, let me just show you the chart I'm talking about here as well. So here's this chart that you want to make sure that you have down for yourself. You want to be able to know the macromolecules, the elements associated with them, the building blocks, or what their building blocks are called, the function. I would say at least know 1 or 2 functions, and then some comments, whatever extra notes you want to write about it. Okay, folks? And our goal is to go through these macromolecules and get details about them. Okay, let's start off with the first one, which is good old carbohydrates. So carbohydrates are going to be sugars and starches. That means when you're eating your bread, when you have sugar in your coffee, all those things are going to be processed as carbohydrates in your body. Now, carbohydrates are going to contain 3 types of elements: carbon, hydrogen, and oxygen. Okay? And usually the hydrogen and oxygen are in a 2-to-1 ratio. So for every 1 oxygen, you know you're going to have 2 hydrogen. That's what that 2-to-1 ratio means. Some of these details, like the 2-to-1 ratio, you might not want to remember, but again, the goal now is for you to just kind of listen and think about what functions you want to remember and what details you want to remember about the carbohydrate. Another thing about carbohydrates is that there's 3 different classes. There's a monosaccharide, disaccharide, and polysaccharide. And so you might be like, wait a minute, I thought we were talking about monomers and polymers. Well, when it comes to carbohydrates, we change the name a little bit because we are talking about sugars. So mono still means one, di means two, poly still means many. But now we talk about saccharides. Saccharide means sugar. So this is saying you're only dealing with one single sugar. These are going to be two sugars, and these are many sugars. So if I was thinking of the monomer here, the monosaccharide would be my monomer, and the polysaccharide would be my polymer. Okay, folks, so that's one of the main things you want to know. What is the monomer? What is the actual polymer, or whatever version of those details we have for lipids? Let's talk about each one of these three classes. First things first, monosaccharides. Monosaccharides, again, are simple sugars. They usually contain 3 to 7 carbons. And so we kind of ask ourselves, well, let's get a general formula for this. So we have CH2O. As you can see, we have the 2-to-1 ratio for the oxygen. Okay, and then we have the little n. That little n means the number of carbon atoms. So usually the number of carbons can change. That's the reason why we have this formula. 3 to 7 carbons. Do you need to know the formula? It's up to you about how you want to remember this information. Now, monosaccharides are the monomers specifically for carbohydrates. So now you want to connect the different verbiage for the macromolecule you're talking about. Excuse me. Why are these so important? Because if I have a simple sugar, like glucose, well, didn't we talk about cell respiration? Yes, we did. So glucose and oxygen can form, as we know, CO2 and water. What else? Energy. What kind of energy? ATP energy. So if I know that monosaccharides are simple sugars like glucose, then I know that this can be a primary source, and this is what this means, primary source of quick energy. E for energy. Okay, folks? Meaning you can get ATP quickly if you have a good amount of glucose around. You don't have to wait around to get energy. It's going to be quick because it's already going to be monosaccharides. It's already going to be that simple sugar that can easily go into cell respiration. Okay, folks? Here's some other monosaccharides. We have ribose, deoxyribose, meaning RNA and DNA. So yes, these sugars are what make up nucleotides when we start talking about nucleic acids. And of course, good old glucose. Next, we have disaccharides and polysaccharides. So disaccharides we know are going to be 2 sugars. And usually they're too large to pass through the cell membrane. So again, they would have to be having broken down a little bit further in order to have that be glucose and be able to cross easier. Okay, folks? Some important ones will be like sucrose, maltose, lactose, as we can see in milk and different foods that we have. And again, we use dehydration synthesis. That's going to be the key word to help join 2 monosaccharides to build this disaccharide. All right, folks? Now let's get into our polysaccharides. So as you can see, we're just going through the details of carbohydrates. The goal is you want to know what the monomer is, the different types of elements. You want to know some functions, okay? Now, polysaccharides, again, are polymers of monosaccharides. Here's an example. We have a bunch of monomers that are going to be coming together through that dehydration synthesis to form these long chains. Disaccharides is 2, monosaccharides is 1. Okay? So some important polysaccharides are starch. And this is how plants are going to store their carbohydrates. And of course, we have long ones called glycogen. And these polysaccharides are going to be stored in our liver and our muscles. Excuse me. And so they're not that soluble, so you're not going to be using them 24/7. They're going to be used when they're needed. So for example, if the blood sugar is low, you might be using stored glycogen to make sure that sugar is released into the blood, for example. Okay, folks? So yes, glycogen can be seen as short-term storage, usually in the muscle and liver. Another beauty about carbohydrates is that they also can help with cells. So on the actual surface of a cell, sometimes they have these little glycoproteins and glycolipids that can help with cell recognition and also cells talking to one another, like signaling one another as well. So there's a lot of pretty cool ideas when it comes to carbohydrates and different functions. Now let's go ahead and go to our chart. So as you can see here, we have carbohydrates. The different elements are carbon, hydrogen, oxygen. I didn't even put the ratio for here. Building blocks will be monosaccharides, disaccharides, and of course polysaccharides. But really the true monomer is going to be a monosaccharide. The polymer is a polysaccharide. Function, primary source of cellular energy. Could I have said short-term storage for glycogen? Could I have said cell recognition and signaling? Yes. And then some comments, it's going to be stored as glycogen in humans. Here's a ratio. Really comments, you can write any notes you want that you want to add. And that can come from the book, that can come from the active reading guide, that can come from the lecture, or whatever you think in your mind. Anything that's going to help you figure that piece out. Let's move on to lipids.